Welcome everyone to our lecture on primary production. So I have all along been saying primary production, mentioning autotrophy, the primary producers, the phytoplankton, and the fact of the matter is that primary production and autotrophy are essentially interchangeable words. I'm going to explain that in a little bit more detail in this lesson. Before we move on to the actual players in the ocean that do this primarily. So what is primary production? In our brief discussions about biogeochemistry, I tried to drive home that what we're really studying is how all elements on Earth move, are converted from inorganic materials or non-living to organic materials, which are living, and in one form or another for specific groups of organisms to be able to use them, or for them to be able to move in and out of certain pools. And what primary production does is take care of that portion of the cycle that turns inorganic or non-living things into organic or living things. This is carbon fixation, essentially, is what we're talking about, especially for the purposes of this class. There are many cycles for all the elements. Each element, I should say, and even some compounds as a whole, have their own particular cycle going from inorganic to organic. But almost all things can be traced back to carbon, and it's another thing I've tried to drive home, and carbon is the main example we use as our biogeochemical cycle. But primary production, the main idea of primary production, its great importance, is carbon fixation. And that's just the term we give to this, turning inorganic material into organic. Inorganic carbon, IC, to organic carbon, or OC. Right? And what we're primarily focusing on in this course, and really this is what's of global importance, is turning CO2, so a carbon source that nobody else can use other than a primary producer, and turning it into an organic molecule, such as a sugar or a carbohydrate molecule, that all other organisms can use it. So that a compound that all other organisms can use, I should say. So in other words, without the autotrophs fixing CO2, doing carbon fixation, all other organisms, essentially all consumers, would not exist. So what we want to do, or at least what I want to do for you guys here, is break down the two major types of autotrophy. Okay, so the guys that do this are the autotrophy. And the two major types are photosynthesis and chemosynthesis. And so if we look back really briefly at this table that I created in our metabolism lesson, we said that you could either be an autotroph, which means you make your own organic materials from inorganic ones, or a heterotroph, which means you consume organic carbon that's already been made for you. And then you can use light or chemical bond breaking as the energy source for doing these types of metabolisms. And when you follow these across, you can be either a light self-feeder or a light-using consumer, or you can be a chemical bond-breaking self-feeder or a chemical bond-breaking consumer. But when we're talking about in primary production is these guys right here, the autotrophs. So you can either be a photoautotroph or a chemoautotroph. So that's photosynthesis or chemosynthesis. Let's start with chemosynthesis. Chemosynthesis is by far, or I should say, is definitely the minor form of autotrophy, especially in the ocean. It does happen to great extents in specific environments, one of which we'll go over right now, and is the basis of the food chain in certain ecosystems. And so it is of great importance, but it is minor compared to all primary production that happens in the ocean. And in chemosynthesis, there are a couple of different chemicals that can be used as the electron donor to create the energy that drives this form of primary production. But one of the major ones is this, H2S. Two hydrogens and a sulfur makes hydrogen sulfate. I'm sorry, hydrogen sulfide. And this is the chemical energy comes from this, breaking the bonds of this guy. And if you look at what does this molecule look similar to, and that's H2S. If you put oxygens there, then you have H2O. So it's a similar type of bond breaking as breaking water. And where this primarily occurs is in extreme environments such as hydrothermal vents at the deep sea. We have this volcanism, we have venting of magma materials which are highly toxic and have a lot of chemicals coming out. And so the ecosystem around an environment like this have evolved to consume essentially these chemicals such as hydrogen sulfide, which pours out of here, and use that energy to do carbon fixation, which becomes the basis of the food chain for a hydrothermal vent community. And these are primarily bacteria that can do this in these environments, and they usually live in symbiosis with things like these giant tube worms and clams and other things. They live in symbiosis to detoxify their water, fix this carbon, turn it into carbohydrate and sugars for the rest of the food chain to feed off of. So the second way, of course, is photosynthesis, and photosynthesis is by far the major form of autotrophy in the land or terrestrial environments, but definitely in the ocean. And so the major difference here is breaking water to create the energy to drive the processes here, but the major energy source is of course sunlight, right? So we're capturing the photons from the sun to drive this reaction, and it's the fixation of inorganic carbon in the form of CO2 ultimately into organic carbon, carbohydrate or sugar. And so I throw up this familiar cross section of the ocean to show the zones, because it should be obvious. Where does this primarily occur? Well, anywhere the sunlight can reach, and the majority of the sunlight reaches just the surface layer of the ocean, the epipelagic or the euphotic zone. So at the surface of the ocean we have 100% of the sunlight reaches the surface of the ocean, and then as it travels through the ocean the sunlight gets reduced. And at the bottom of this zone, the euphotic zone, only about 1% of the sunlight reaches there, and that's where almost all of the ocean photosynthesis occurs, is in this thin surface layer. We'll talk more about the layers of the ocean in another lecture. So how do they do this? The whole thing is facilitated by photopigments. We should have learned this a couple of times throughout your pre-college career, your pre-collegiate career, when talking about photosynthesis in land plants. But the cells of even the phytoplankton in the ocean have photopigments, and most of us are familiar with the chlorophylls, especially chlorophyll A. We still use that as a strong indicator of growth of autotrophy, or of what kind of primary production is going on. But there's actually a whole host. There's other chlorophylls. There's chlorophyll B, chlorophyll A. There are different variations on the theme of chlorophylls A's and B's, but there's also totally different kinds of photopigments. One's called carotenoids, one's called phycobilins, and there are variations on these as well. And the main idea, or the take-home thing with these variations on photopigments, is that each one absorbs light at different wavelengths. So if we measure the visible spectrum across here in wavelengths, UV light is down here, but at about 400 and below, and then the highest reds in the spectrum are about 700, and in between is what we can see. And so you can see here, this is the absorption spectrum of chlorophyll A. So it absorbs very high in sort of the high purple to low blue range, but it also absorbs a lot of light in the mid red, so the high orange to mid red range. And whereas chlorophyll B would absorb a little bit differently, a little bit of a shift. It would absorb in here and around here, and the carotenoids and phycobilins, they're all going to absorb in different places. And so each species of phytoplankton, or I should say even other plants, have evolved to have their own set of photopigments so that they can so that not everybody is hogging one wavelength of light, and so they can all do their photosynthesis. And the whole point is to create energy molecules. They harvest the light, they package it, they consume carbon dioxide and water, and they use it to turn their internal machinery and generate things like adenosine triphosphate, ATP, and NADPH, and these are the molecules in all organisms that are responsible for creating energy, so that you can create sugars and store them as carbohydrates or glucose, or burn them, and to maintain your cellular fitness, and to reproduce, and all the other metabolic things that we've talked about so far. And the way that they do this, the photopigments, is it's embedded in a portion of their usually surface of the cells, and the photopigments are embedded in a couple of different places, and the light hits them, and they're able to trap that light energy, use it to split water into oxygen, which is a byproduct that's good for all air breathers, and the hydrogen molecules, and it sets off this chain reaction that generates a lot of energy that hits another set of photopigments that absorb more light, that further drives the energy, and it sets off one metabolic reaction that creates energy, and then hydrogens can be flushed to a different part of the cell and out through what essentially is this tiny little generator. It turns this propeller of a generator here and generates even more energy, and that's what starts the whole metabolic process. That's how the light is trapped and turned into energy. Okay, so that's very brief and basic, but you guys should be familiar with now the two main types, and which one is the largest of, and how they work, of autotrophy, which is essentially primary production. Thanks. See you next time.